I want you to just close your eyes for a second and try to imagine the world in the year 1900.
It's a pretty drastically different picture from
Well, not
today.
completely. I mean, if
Yeah.
you were born in the U.S. right at the turn of that century, your life expectancy at birth was, well, it was roughly 47 years.
Right, 37.
Which is wild to think about.
Yeah.
By the time you reached what we'd now call early middle age, you were, statistically speaking, staring down the end of the line.
Yeah, that demographic shift between then and now is, it's honestly one of the most profound alterations in all of human history.
Seriously.
If you look at demography, there's this thing called the Gompertz-Makeham law of mortality. It basically models the mathematical curve of human death rates.
Okay.
And under those old models, living to 100 in 1900 wasn't just rare, it completely broke the standard curves. Out of, billions of people on Earth, demographers think there were maybe just a few thousand individuals who had ever hit that century mark.
It's just a few thousand. Wow. And, fast forward to today, the average life expectancy in the U.S. has jumped up to 79.
Right.
And in places like Japan, it's 84. Monaco is creeping up near 90. We currently have something like 600,000 centenarians globally.
And the projections for the year 2100 point to around 25 million.
25 million people living to 100.
Mm.
So, the obvious thought is that we just figured it out, right? Like, we somehow unlocked the secret to human longevity in a single century.
Well, no, that is actually a really common misconception.
Wait, really?
Yeah, we didn't unlock some secret to slowing down the fundamental biology of aging. What we experienced was an epidemiological transition.
Okay, what does that mean exactly?
It means the massive gains in life expectancy mostly came from eradicating the stuff that killed young people. We built municipal sanitation systems, we discovered antibiotics, rolled out mass vaccines, made childbirth safer.
I see. So we didn't extend the maximum lifespan. We just...
They basically neutralized infectious diseases, the main thing killing us early.
Right, so we stopped early death, and that just dragged the statistical average way up.
Precisely. We improved life expectancy, which is just an average, but until very recently, we really haven't moved the needle on maximum human lifespan. Lifespan is the absolute biological ceiling for our species.
Which naturally brings us to the ceiling itself.
Yeah.
Jean Camel.
Yes, the famous French case.
Right. The woman who died in 1997 with the verified record. 122 years and 164 days.
The incredible record.
She met Vincent van Gogh when she was a teenager, and she lived long enough to see the internet become a commercial thing. For the longest time, the prevailing wisdom was just that someone like her won the ultimate genetic lottery, right?
Yeah, that her DNA was just fundamentally different from ours. And that assumption basically dictated decades of gerontology research.
Oh, really?
Oh, yeah. Scientists spent years sequencing the genomes of extremely long-lived people, just hunting for this elusive longevity gene.
Like a magic bullet in the DNA.
Exactly. But the data from those studies actually overturned that whole genetic determinism idea, especially these massive studies on twins. The Danish twin registry studies are the famous ones.
What did they find?
They consistently showed that genetics accounts for, at most, 20 to 30% of the variation in human longevity.
Wait, only 20 to 30%?
That's it.
Okay, so if the hard-coded DNA only counts for 20 to 30%, that leaves this enormous variable, like 70 to 80%, just completely up for grabs.
Up to the environment, yeah. It's controlled by environmental inputs, behavioral architectures, and the biochemical reactions those things trigger in our bodies.
And that huge 70 to 80% chunk is exactly what we are mapping out today on this deep dive. We're looking at the exact molecular and sociological mechanisms that allow certain groups of people to just push that biological ceiling.
It's a fascinating area of study.
We're going to examine how environmental stress alters your DNA, why our cellular maintenance systems break down, the massive historical graveyard of failed attempts to biohack these systems.
There are a lot of those.
So many. And we're also going to get into the profound realization that social infrastructure actually dictates our cellular health. Because this isn't just about adding years to your life, it's about adding life to those years, finding practical stuff you can use today.
Exactly.
So to find those environmental inputs, we kind of have to look at the anomalies, right?
Right. If genetics isn't the whole story, geography and environment are the next logical places to look.
Which brings us to the early 2000s. Dan Buettner, working with National Geographic, went out and identified these statistical anomalies. They dubbed them the blue zones.
Yeah, these were specific pockets around the world where extreme longevity wasn't just a fluke, it was the baseline norm.
So what were the zones they found?
They identified 5 primary regions. First, you have the Barbagia region of Sardinia, Italy.
Okay.
And that area holds an unusually high concentration of male centenarians, which is demographically very rare.
Right, because women usually outlive men.
Exactly. Then there's Okinawa, Japan, which historically holds the record for the longest-lived women.
Okay, that's 2.
Third is the Nicoya Peninsula in Costa Rica, boasting the world's lowest rate of middle-age mortality. Fourth is Ikaria, Greece, an island where people essentially forget to die, vanishingly low rates of dementia there.
And the 5th one is actually in the U.S., right?
Yeah, an unexpected outlier. A community of Seventh-day Adventists in Loma Linda, California.
Okay, let's unpack this for a second.
Yes.
Because looking at that list from a strictly environmental standpoint is baffling.
How so?
Well, you've got isolated mountainous shepherds in Italy, coastal fishermen in Japan, and then a suburban religious community in Southern California. They don't share a climate.
No.
They don't share a genome. They don't even share a diet. I mean, the Adventists are mostly vegetarian. The Sardinians are eating sheep-smoked cheese. And the Okinawans are heavily reliant on fish and root vegetables.
Right.
There's no single miraculous superfood that connects all these people.
And honestly, that lack of a shared specific ingredient is exactly what makes the blue zones so scientifically valuable.
Because it proves it's not just about eating goji berries or whatever.
Exactly. It forces researchers to look past the superficial details. It's not about whether they eat fava beans or purple sweet potatoes. It's about the underlying biological pathways those environments activate. Researchers managed to extract a shared architectural framework of living across all 5 zones. It's a set of principles that ultimately dictates how their cells process energy and handle damage.
Okay, so let's strip away the geography and look at that architecture. I know researchers compiled these into something they call the Power 9. Let's start with the mechanical demands they place on their bodies.
Movement.
Right, because in Western wellness culture, we completely compartmentalize movement. We sit at a desk for 9 hours straight, and then we drive to a gym for 45 minutes to just artificially stress our cardiovascular systems.
And the physiological response to that gym model is very different from what we see in the blue zones. They engage in what exercise physiologists call non-exercise activity thermogenesis.
Neat.
Right? Neat. Their environments demand constant, low-level physical exertion.
Like what kind of stuff?
Well, they're kneading bread by hand, navigating steep, uneven terrain just to get to a neighbor's house, gardening manually, walking to communal gatherings. There's no scheduled gym time.
So instead of a massive acute spike in heart rate followed by prolonged sitting, their bodies are experiencing a constant low-grade mechanical load all day long.
Exact.
How does that actually change the cellular environment?
It drastically alters glucose disposal and insulin sensitivity.
Okay, break that down for me.
Sure. When you are constantly moving, your muscle cells are continuously upregulating glucose transporters, specifically these proteins called GLUT4, to the surface of the cell.
To what?
To pull sugar out of the bloodstream to use for energy. So by doing this constantly, you are preventing the chronic hyperinsulinemia, the high insulin levels that drives systemic inflammation in Western populations.
Because the sugar never gets a chance to just sit around and cause trouble.
Right. Their movement is inescapable. It is literally built into the topography of their daily existence.
That makes so much sense. And what fuels that movement is just as critical.
We mentioned they don't share one specific food, but they do have a shared macronutrient philosophy, right?
They do. It is an overwhelmingly plant slant diet.
Plant slant, I like that.
Yeah, mostly plants. Beans are usually the cornerstone, fava, black, soy, lentils. Meat is incredibly rare. It's treated more as a celebratory addition, maybe a few times a month, rather than a daily staple.
But the most fascinating mechanism here, to me at least, isn't just about avoiding meat. It's about the caloric load itself.
Ah, yes.
The Okinawans have this practice called harahachibu.
Right. It's a Confucian teaching. Yeah, they literally repeat it before meals as a reminder to stop eating when they feel about 80% full.
And from a molecular gerontology perspective, that mantra is arguably the most potent behavioral intervention they practice.
Really? Just stopping at 80%.
Yes. By consistently stopping before they're stuffed, they are inducing a mild, sustained state of caloric restriction.
And I know caloric restriction is like the golden child of longevity research in animal models. But why is a slight caloric deficit so incredibly powerful at the cellular level?
It comes down to a fundamental cellular energy sensor. It's an enzyme called AMPK.
AMP-activated protein kinase.
That's the one. When your cells sense a scarcity of energy, meaning there's a low ratio of ATP available, AMPK gets activated.
Okay.
This enzyme is essentially a master metabolic switch. When it turns on, it signals the entire cell to stop building new tissue and instead focus heavily on repair and maintenance. It triggers a process called autophagy.
Autophagy.
Cha.
Auto meaning self, phagy meaning eating, so the cell starts eating itself.
Essentially, yes. Think of it as a cellular recycling program.
Oh.
The cell starts hunting down its own damaged proteins, misfolded enzymes, old dysfunctional organelles. It breaks them all down into basic amino acids to use for fuel or to make essential repairs.
Oh, wow. So it's literally cleaning house.
Exactly. But here's the catch. When we are constantly fed, when we eat until we are 100% full, multiple times a day, AMPK stays completely suppressed.
Because the body thinks, hey, times are good, let's grow.
Right. We are constantly in a state of growth and storage. And so that internal garbage collection system, the autophagy, just shuts down.
And then what happens?
The debris accumulates. All those misfolded proteins and damaged organelles build up. And that leads directly to cellular dysfunction and aging.
So Hara Hachibu isn't just a trick to stay lean. It's a daily, culturally mandated trigger for deep cellular cleanup.
It absolutely is.
That is wild. But the blue zones aren't just defined by diet and movement. The social and psychological architectures are where the mechanisms get, honestly, deeply complex.
Very much so.
In the West, we largely view stress as a psychological nuisance. Like, oh, I'm stressed, I need a spa day. But in these zones, they view the shedding of stress as a daily, non-negotiable necessity.
Because chronic psychological stress translates into a highly destructive biochemical reality.
Walk me through that. What happens when we're stressed?
When the brain perceives constant low-grade threats, and nowadays that's financial insecurity, social isolation, just overwhelming modern schedules, the HPA axis remains locked in an active state.
The hypothalamic pituitary adrenal axis.
Yes. And when that's constantly on, you are continuously bathing your tissues in glucocorticoids, primarily cortisol.
And cortisol is good in an emergency, but over the long term, it's toxic.
Highly toxic. It degrades immune function, it increases your blood pressure, and it drives a systemic inflammatory response throughout the whole body.
So how do the blue zones handle this? They must get stressed too.
They do, but their cultures have built-in pressure valves to physically downregulate that HPA axis every single day.
Like what?
Well, the Sardinians have an afternoon gathering with local wine.
Sounds great.
Right? The Icarians mandate a mid-afternoon nap, which physically resets cortisol rhythms, and the Adventists enforce a strict Sabbath.
Where labor and commerce are totally forbidden.
Exactly. They entirely remove the primary vectors of modern stress for 24 uninterrupted hours every week.
Man, we could all use that. And this directly ties into their social networks, which honestly might be the most robust environmental input of all.
The social aspect is massive.
Like in Okinawa, they have this concept called the moai.
Yes.
It's this deeply committed group of 5 friends, usually established in childhood, and they just share financial, emotional, and physical burdens for life.
They essentially distribute the allostatic load, the physiological wear and tear of stress, across multiple nervous systems.
That is such a cool way to phrase it, distributing the load across nervous systems.
And we have concrete data on how health behaviors propagate through these kinds of networks. There's a physician and sociologist named Nicholas Christakis who has done incredible network mapping on this.
Oh, right. Showing that behaviors are actually socially contagious.
Yes.
Like, if your friends start smoking, your statistical probability of smoking just skyrockets. But his research goes beyond just conscious choices, doesn't it? It affects baseline biology.
It really does. Christakis' work demonstrates that even biological outcomes like obesity spread through social ties.
Wait, really? Obesity itself is contagious.
Statistically, yes. If a close friend becomes obese, your chances of becoming obese increase by over 50%.
Even if they don't live near you.
Even if they live hundreds of miles away. The social network dictates your normalized standard of behavior.
That is mind-blowing.
Yeah.
It's like a compounding effect for your health.
Exactly. In a moai, or a multi-generational Sardinian household, the default normalized behavior is intense social support, natural movement, and plant-based restricted eating.
So you don't have to exert all this massive individual willpower to be healthy. The environment just pulls you into health. Modern society treats health like this solitary, miserable pursuit. Going to the gym alone, dieting alone.
But in blue zones, health is an inescapable environmental default.
Okay, so we have this overarching behavioral architecture. But we need to translate that behavior into actual molecular biology.
Uh-huh.
How does a daily walk with your moai or a mild caloric restriction physically stop the clock inside our bodies?
To understand that, we have to dive into the hallmarks of aging. Let's start with the cellular replication limit.
Okay, let's do it.
Back in the 1960s, a researcher named Leonard Hayflick discovered that human cells cannot divide infinitely. There is a hard limit.
The Hayflick limit.
Right. It's usually around 50 divisions for a normal human fetal cell. But the mechanism behind why this limit existed wasn't fully understood until researchers identified telomeres.
Okay, I've always pictured DNA as this incredibly long, fragile string of data. And the telomeres are the repeating, non-coding sequences of DNA at the very tips of our chromosomes. Yeah. Are they functioning essentially like the plastic aglets at the end of a shoelace? Like stopping the vital genetic code from fraying during replication?
That is the exact mechanical function. Because of how DNA polymerase, which is the enzyme that copies our DNA, because of how it works, it physically cannot replicate the very, very end of the strand. So with every single cellular division, a tiny piece of that telomere is lost. The plastic aglet gets shorter and shorter.
And eventually it's gone.
When the telomere reaches a critical shortness, it triggers a massive DNA damage response. The cell senses that its actual genetic integrity is at risk, so it halts all division to prevent mutating into a cancer cell.
And this halted state is what biologists call cellular senescence.
Yes.
The cell doesn't die, but it stops functioning normally. I've heard gerontologists refer to these as zombie cells.
And zombie cell is a great term because it captures the parasitic nature of senescence. A senescent cell refuses to undergo apoptosis, which is programmed cell death.
It just lingers.
It just lingers in the tissue. But more critically, it undergoes a profound shift in how it behaves. It adopts what we call the senescence-associated secretory phenotype, or SSP.
Secretory phenotype, meaning it's actively spitting something out into the surrounding tissue.
Exactly. It begins secreting a highly toxic cocktail of pro-inflammatory cytokines, chemokines, and tissue-degrading proteases.
Wow.
It is basically screaming a chemical alarm signal into the surrounding microenvironment, and this paracrine signaling actively damages neighboring healthy cells.
It's like it's spewing acid on its neighbors.
Pretty much. It accelerates their path towards senescence too and drives massive local and systemic inflammation.
So one zombie cell makes more zombie cells.
Yes, and the accumulation of these SSP-secreting cells over decades is a massive driver of things like osteoarthritis, atherosclerosis, and neurodegeneration.
Okay, so shortening telomeres create zombie cells, which secrete inflammation, driving systemic aging. Now, we do have an enzyme called telomerase that can actually rebuild these telomeres, right?
We do.
But it's mostly turned off in adult somatic cells. Is there a way to naturally upregulate it? Because this is where the work of Elizabeth Blackburn gets genuinely wild.
Yes, she won the Nobel Prize for her telomere research, and her work provided the biochemical bridge between psychology and molecular biology.
What did she do?
Her team studied mothers who were caring for chronically ill children. So women in a state of profound, unrelenting psychological stress.
Right.
They found that the higher the mother's perceived stress, the shorter her telomeres were, and the lower her telomerase activity was.
Oh my God.
The stress was physically accelerating their cellular aging by suppressing that maintenance enzyme.
But the inverse was also true, right?
Yeah.
And this is the part that sounds like actual magic.
It really does.
Her later studies suggested that specific psychological interventions like intensive mindfulness retreats or loving-kindness meditation were associated with increased telomerase activity.
Yes, they were.
How does sitting quietly and regulating your breathing fundamentally alter an enzyme interacting with your DNA? Like, how does that mechanical bridge work?
It goes right back to the HPA axis and the cortisol we discussed earlier. Your genome is not sitting in some isolated vault. It is highly responsive to the chemical bath it sits in.
Okay.
When you engage in deep vagal nerve stimulation through meditation or through profound social connection, you drastically lower the concentration of inflammatory cytokines and stress hormones in your blood.
You change the bathwater.
Exactly. You remove the biochemical suppression of telomerase. You are changing the chemical signaling environment, which allows the cell to resume its baseline maintenance protocols and rebuild those telomeres.
That is just, it's amazing. Behavior alters the chemical bath, which alters the enzyme, which protects the DNA. It makes perfect mechanical sense.
It does.
But the DNA is only one part of the cellular equation. What is happening to the energy producers of the cell, the mitochondria?
Well, as we age, mitochondrial dysfunction becomes another primary driver of decay. Mitochondria generate ATP through something called the electron transport chain. But this process is inherently messy.
Messy Al.
It produces a byproduct called reactive oxygen species, commonly known as free radicals.
Right, I've heard of free radicals. These are molecules missing an electron, so they violently rip electrons from surrounding structures, damaging proteins and DNA. It's oxidative stress.
Yes. I think of it like a poorly maintained nuclear reactor where the containment fields are failing and it's allowing localized radiation to slowly degrade the surrounding facility.
That is a highly accurate mechanical representation. In our youth, our cells produce robust endogenous antioxidants to neutralize those free radicals. But as the mitochondria age, they become less efficient. They produce significantly more free radicals, which overwhelms those containment fields.
So how do we fix the reactor? This is where the specific type of physical movement comes into play, isn't it? Yes. While the blue zones rely on that low-level NEAT, modern exercise physiology shows us that high-intensity interval training HIE can actually reverse this mitochondrial decay.
Wait, how does stressing the system with high-intensity exercise fix a broken reactor? That feels counterintuitive.
It activates a pathway controlled by a protein called PGC1 alpha. When you rapidly deplete ATP during a high-intensity sprint, the cell panics.
Because it's running out of gas.
Exactly. It realizes it doesn't have the energy infrastructure to survive that kind of sudden demand. So PGC1 alpha enters the nucleus and triggers a process called mitochondrial biogenesis.
Biogenesis, making new ones.
It forces the cell to build brand new, highly efficient mitochondria while simultaneously increasing the expression of those endogenous antioxidants.
You are literally forcing the cell to upgrade its power grid by demonstrating that the current grid is insufficient.
That's exactly what's happening.
This adaptability is just incredible. And that leads us to the master control board of all these processes, the epigenome.
Yes, the epigenome. We used to view the genome as destiny, but the genome is really just the raw hardware code.
Right.
The epigenome is the software. It dictates which parts of that code are actually read and executed by the cell.
How does it do that?
Primarily through something called DNA methylation. It's the addition of tiny chemical tags methyl groups to specific regions of the DNA. These tags physically prevent transcription factors from accessing that gene, effectively silencing it.
So it turns genes on and off. And as we age, does this methylation process just become chaotic?
Highly chaotic. Genes that should be silenced, like pro-inflammatory genes, they lose their methyl tags and get turned on.
Oh, bad.
Very bad. And genes that should be active like tumor suppressors, they gain methyl tags and get turned off.
Double bad.
Right. This predictable degradation is actually what allowed a researcher named Steve Horvath at UCLA to create the epigenetic clock.
Oh, I've heard of this.
By looking at the methylation patterns of specific locations on the genome, he can calculate a person's biological age, and that often differs wildly from their chronological age.
So a person might be 60 chronologically, like on their driver's license, but their software is so degraded that their epigenetic clock says their cells are 75.
Exactly.
But the behaviors we've discussed, the caloric restriction, the constant movement, the stress management in the blue zones, these effectively preserve those methylation patterns.
Yes, they preserve the integrity of the software, they keep the correct genes silenced and the necessary genes active.
Which naturally brings us to the ultimate human outliers, the supercentenarians.
Ah, yes, the 110s and older.
Right, because if environmental factors preserve our biology, what happens when someone has an optimal environment and a highly unique genetic baseline? What is mechanically different about a supercentenarian?
There was a landmark 2020 study published in the Proceedings of the National Academy of Sciences by Hashimoto and his colleagues. They took a deep dive into the immune profiles of supercentenarians.
And what did they find?
They performed single-cell transcriptomics on their blood and found something highly unusual. These individuals possessed heavily expanded populations of a very specific immune cell, CD4 plus cytotoxic T lymphocytes.
Okay, let's break that down, because normally CD8 T cells are the primary cytotoxic ones, right? They're the assassins of the immune system that kill virus-infected or cancerous cells.
Correct.
And CD4 cells are usually just the helper cells. They just organize the immune response. They don't do the killing.
Exactly. But in supercentenarians, these CD4 helper cells have evolved into a highly aggressive cytotoxic phenotype.
They became assassins.
They essentially acquired the ability to directly kill target cells. And the current working hypothesis is that this expanded army of CD4 cytotoxic cells acts as a highly specialized cellular hit squad.
A hit squad for what?
They are phenomenally efficient at hunting down and eliminating those senescent zombie cells we discussed earlier.
Oh, wow.
Yeah, so they prevent the accumulation of that SSP inflammatory cascade.
So the supercentenarian secret isn't that they magically don't produce damaged cells. It's that their immune system recognizes and annihilates those damaged cells with ruthless efficiency before they can secrete inflammation.
Exactly. They have an incredibly active cellular janitorial staff doing constant maintenance. And this specialized immune clearance leads to a demographic phenomenon called the compression of morbidity.
Compression of morbidity, what does that look like?
Well, when people think of living to 110, they usually envision spending 30 years in a state of frail, agonizing, bedridden decline.
Yeah, which sounds awful.
But the Gompertz curve at extreme ages shows the exact opposite.
Really? They don't spend 20 years sick.
No, they stay highly functional for a century, and then they experience a very rapid, compressed decline right at the very end. Their health span basically equals their lifespan.
That is the dream right there. Now, we mentioned earlier that genetics only accounts for 20 to 30% of longevity. But for these supercentenarians, there are specific genetic variants at play. We need to talk about APOE and FOXO3.
Yes, those are key. The apolipoprotein E, or APOE gene, is primarily involved in lipid metabolism, how your body transports cholesterol and fats.
Okay.
It comes in 3 major alleles, epsilon 2, epsilon 3, and epsilon 4. If you inherit the epsilon 4 variant, your risk for late-onset Alzheimer's disease and cardiovascular atherosclerosis just skyrockets. It fundamentally alters how your brain clears amyloid beta plaques.
It's the bad one. But the supercentenarians frequently possess the epsilon 2 variant, right?
Yes. The epsilon 2 variant is highly protective, it enhances lipid clearance, and appears to really protect the neurological infrastructure.
But FOXO3 is arguably even more fascinating to me because it acts as a master regulator of longevity across multiple species, not just humans.
It does. It's a transcription factor.
Meaning its entire job is to enter the cell nucleus and turn other genes on or off. What specific genes is FO3 targeting?
It upregulates a vast array of protective genes. It increases the expression of DNA repair enzymes. It upregulates genes involved in autophagy to clear out that cellular debris.
There's that autophagy again?
Exactly. And crucially, it upregulates antioxidant enzymes like superoxide dismutase 2, which neutralizes those free radicals leaking from the mitochondria.
Wow.
So when someone inherits a specific protective variant of FOXO3, their cells are hyper-responsive to damage.
Okay, here is the crucial pivot, though. This is the absolute core of the longevity puzzle. Because a listener might hear about FOXO3 and think, well, I didn't inherit the rare variant, so I don't have that superpowered DNA repair, I'm out of luck.
But they aren't out of luck.
Right. The presence of the rare gene variant isn't the only way to activate the pathway, is it?
Not at all. And this is the profound biological reality of the blue zones.
Yeah.
FOXO3 is highly sensitive to environmental inputs.
Like what?
Specifically, it is activated by the exact same nutrient-sensing pathways we discussed earlier, like AMPK.
Oh.
When the Okanons practice hara hachibu, or when someone engages in intermittent fasting, the resulting drop in insulin signaling and the activation of AMPK directly phosphorylates FOXO3.
Turns it on.
It sends it straight into the nucleus to turn on those exact same longevity and repair genes.
So through targeted behavioral stress like caloric restriction or vigorous movement, we can manually force our standard issue FOXO3 genes to mimic the biological behavior of a supercentenarian's rare genetic variant.
You are literally hacking the software.
We are inducing the transcription of longevity genes through environmental pressure. That is unbelievable.
It's very empowering biology.
But okay, if the mechanisms are this clearly mapped out, AMPK, MTOR, FOXO3 telomerase, it raises the most obvious question in modern medicine.
Why isn't there a pill?
Exactly. Why haven't pharmaceutical companies just isolated a molecule that perfectly trips these biochemical switches? Why do I have to eat fava beans and walk up mountains? Let's wander through the graveyard of anti-aging pharmacology, starting with the massive resveratrol boom.
Oh, the resveratrol boom. The early 2000s were completely dominated by resveratrol. It's a polyphenol found in the skin of red grapes.
Right.
Researchers, notably David Sinclair's lab, found that resveratrol could activate a class of proteins called sirtuins, specifically SRT1.
What does sirtuins do?
They're NAD-dependent enzymes that also regulate cellular health, DNA repair, and mitochondrial function. And in yeast, worms, and obese mice, massive doses of resveratrol dramatically improved health and extended lifespan.
And the media immediately translated that to Drinking red wine cures aging. I remember the headlines.
Which was a catastrophic misinterpretation of pharmacokinetics.
Let's hear the bad news.
The bioavailability of resveratrol in humans is incredibly low. It is rapidly metabolized and excreted. To achieve the blood concentrations used in those successful mouse models, a human would have to consume liters upon liters of red wine daily.
You die of cirrhosis long before experiencing any SIRT1 activation.
Exactly. And when highly concentrated synthesized resveratrol formulations were finally put through rigorous human clinical trials, the longevity and metabolic benefits largely evaporated.
So it just didn't translate from mice to humans.
Yeah. The human biological network was simply too complex for a single isolated polyphenol to override the system.
Okay, what about caloric restriction? We know manipulating the AMPK and MTOR pathways works. Can we pharmacologically mimic harahachibu?
We can try. We have drugs like rapamycin.
I've heard of rapamycin.
It effectively inhibits the MTOR pathway, essentially tricking the cell into thinking it is starving. So it induces autophagy without actual caloric restriction. In mice, rapamycin is the most successful pharmacological life extender we currently have.
I feel a butt coming.
But the human translation is fraught, because MTOR isn't just an aging pathway, it's fundamentally a growth and immune pathway.
Right.
Rapamycin is primarily used clinically as an immunosuppressant for organ transplant patients. If you chronically suppress MTOR in a healthy human to prevent aging, you simultaneously suppress their adaptive immune system.
Making them highly vulnerable to lethal infections.
Yes. You also blunt muscle protein synthesis, leading to sarcopenia, which is severe muscle loss. And muscle loss is an independent predictor of mortality in the elderly.
It's the concept of antagonistic pleiotropy. The biological switch that promotes youthful growth and immunity is the exact same switch that drives cellular senescence and cancer in old age.
Exactly.
You can't just turn it off without paying a massive physiological toll. And this was disastrously evident in the craze for growth hormone replacement, right?
Oh, that was a dark chapter. In the 1990s, anti-aging clinics began prescribing recombinant human growth hormone to wealthy clients.
Because it makes you feel young.
In the short term, the results looked miraculous. It increased IGF-1, or insulin-like growth factor 1 levels. Patients saw increased lean muscle mass, reduced body fat, thicker skin, and a profound surge in energy. They literally felt 20 years younger.
But IGF-1 is the ultimate pro-growth signal. It tells every cell in the body to proliferate.
Including the microscopic precancerous cells that a normal aging immune system was quietly holding in check. The long-term epidemiological follow-ups were grim. Artificially elevating growth hormone and IGF-1 significantly accelerated tumor progression and insulin resistance. The intervention traded a brief illusion of physical youth for an accelerated, fatal pathology.
That is terrifying. The ultimate lesson from this graveyard is that the human organism is a highly integrated, finely tuned network.
It is.
Trying to biohack aging by hyperstimulating a single pathway, whether it's SIRT1, MTOR, or IGF-1, is like trying to fix a complex symphony by just making the violins play as loud as possible. It completely destroys the harmony of the system.
That's a perfect analogy. The Blue Zones achieves their longevity not by biohacking a single pathway, but by providing a massive holistic orchestra of moderate environmental inputs.
Movement, plants, stress reduction.
Right. They gently nudge the entire biochemical network toward maintenance rather than growth.
But this brings us to a massive sociological roadblock.
Right.
We've defined the optimal environmental inputs, but we have to acknowledge that access to these inputs is highly restricted in the modern world.
It's the reality of modern society.
If we step out of the cellular biology and into public health, we hit the wall of social determinants.
And the socioeconomic disparity in longevity is arguably the most pressing public health crisis in the developed world.
How bad is the gap?
If you look at mortality data in the United States, the gap in life expectancy between the top 1% of income earners and the bottom 1% is staggering. For men, it is a gap of nearly 15 years.
15 years. That is insane. That is the equivalent difference in life expectancy between the United States and a developing nation without modern sanitation, existing within the exact same borders.
It's a stark reality. Public health researchers often say that your zip code is a far more accurate predictor of your lifespan than your genetic code.
How exactly does poverty translate into accelerated cellular decay? Like mechanically?
It operates through that concept of allostatic load we mentioned, the cumulative wear and tear on biological systems caused by chronic environmental stress. It is not just about the inability to purchase organic vegetables at a farmer's market. It is the physiological toll of navigating a food desert where the only accessible calories are metabolically disastrous ultra-processed foods.
And environmental factors, too.
Yes. It is the systemic exposure to environmental toxins, lead pipes, and industrial air pollution that drive chronic inflammation.
And it's the psychological stress of systemic inequality, the chronic threat of eviction, the reality of discrimination, the inability to access preventative medical care.
All of those psychosocial stressors keep the HPA axis permanently engaged. The body becomes resistant to cortisol.
Resistant, meaning it stops working.
The glucocorticoid receptors downregulate, meaning the body's natural anti-inflammatory brakes basically stop working.
Wow.
The chronic systemic inflammation physically degrades the telomeres, induces cellular senescence, and accelerates the epigenetic clock. Poverty is literally mechanically aged into the tissues.
And interacting with this economic disparity is an even broader cultural pathology, the epidemic of loneliness.
This is a huge factor.
We talked about the protective nature of the Okinawan Moai, that tight-knit group of lifelong friends. What happens to the cellular biology when that social network is entirely absent?
The research on isolation is deeply unsettling. The psychologist Julianne Holt-Lundstad conducted a massive meta-analysis encompassing millions of individuals. She examined the mortality risk of social isolation.
Yeah.
She found that chronic loneliness and social isolation increased the risk of mortality by roughly 30%.
The statistic from her work that always floors me is her comparison to smoking.
Yes, her data suggests that the mortality impact of prolonged social isolation is functionally equivalent to smoking 15 cigarettes a day.
15 cigarettes a day just from being lonely.
It is more lethal than clinical obesity or physical inactivity.
But how? How does the feeling of loneliness, which is a strictly emotional state, cause physical cardiovascular disease or immune failure?
It operates through a highly specific genomic pathway called the conserved transcriptional response to adversity, or CTRA.
CTRI.
Evolutionary biologists hypothesized that for early hominids, social isolation meant imminent physical danger, predation, or starvation.
Because if you're away from the tribe, you're probably going to get eaten by a lion.
Exactly. So when the modern brain perceives chronic isolation, it still assumes you are about to be wounded. It signals the immune system to upregulate genes that cause inflammation, to prepare for bacterial infection from a physical wound.
Oh my God.
And simultaneously, it downregulates the genes responsible for fighting viral infections, since you are no longer around other humans to catch a virus from.
It physically alters gene expression to prepare for physical trauma that never actually comes, leaving the body soaking in unnecessary damaging inflammation while simultaneously degrading its antiviral defenses.
Exactly. The feeling of loneliness is an evolutionary alarm bell that fundamentally scrambles your immune software.
This honestly makes me view the entire longevity industry with profound skepticism.
It really reframes things.
We have a culture spending billions of dollars individually on cold plunges, hyperbaric chambers, and boutique NAD plus IV drips. But the data unequivocally states that if we want to increase human lifespan, we shouldn't be funding longevity clinics. We should be funding urban planning,
Absolutely.
designing walkable cities, ensuring neighborhood safety, funding community centers, and reliable bus systems so the elderly aren't isolated in suburban sprawl. A library might do more for public lifespan than a new vitamin. Infrastructure is the most powerful longevity intervention we have.
The empirical data supports that conclusion entirely. If the environment dictates the biological pathways, then civic engineering, economic equity, and social architecture are the ultimate upstream regulators of human cellular health.
We've covered the cellular engines, the genetic software, the pharmacological failures, and the socioeconomic reality. But there's one final, deeply personal metric that ties all of this together: the sheer will to live.
The biology of purpose.
Right. In the blue zones, this concept is highly formalized. The Nicoyans in Costa Rica call it plan de vida. The Okinawans call it ikigai.
Literally translated as the reason for which you wake up in the morning.
And we tend to view this through a highly romantic, philosophical lens, like having a hobby, feeling fulfilled. But the demographic research points to something far more structural. Let's look at that 2014 study by Patrick Hill and Nicholas Turiano in Psychological Science.
That's a crucial study. Hill and Turiano tracked over 6,000 participants over a 14-year period.
A good sample size.
They found that individuals who scored high on assessments of life purpose had a significantly lower risk of mortality across all age groups.
And the critical detail here is that this survival advantage held true even after controlling for baseline physical health, psychological well-being, and socioeconomic status.
Meaning it wasn't just that healthy, rich people felt more purposeful. The abstract sense of purpose itself was exerting an independent, protective biological effect.
How does a philosophical construct shield a cardiovascular system?
Well, there are behavioral mediators, obviously.
Right.
People with purpose are more likely to seek preventative health care, maintain regular sleep architecture, and comply with medical advice because they feel their future has utility.
They want to be around for it.
Right. But there is also a profound neuroendocrine component.
This relates to the research by Steve Cole on the different types of happiness, doesn't it? The difference between just feeling good and feeling meaningful.
Yes. Cole's research distinguishes between hedonic well-being, which is the happiness derived from immediate pleasure or consumption, and eudaimonic well-being.
Eudaimonic. That's
Yeah.
the happiness derived from a sense of meaning and contribution to a greater good.
Exactly. Cole found that people with high eudaimonic well-being showed significant downregulation of that inflammatory CTRA gene profile we just discussed.
The loneliness pathway.
Right. Conversely, people who only experienced hedonic, pleasure-seeking happiness actually showed an increase in inflammatory gene expression, basically similar to people experiencing chronic stress.
That is absolutely profound. The cells can actually differentiate between the biochemical signature of a meaningful existence and the biochemical signature of simple pleasure-seeking.
It appears so.
Ikigai is not a philosophical luxury. It is an active genomic command telling your immune system to downregulate inflammation because the organism is required for a long-term structural purpose.
The organism requires a biological signal that its continued existence is necessary. Without that signal, the pathways of maintenance and repair begin to yield to the pathways of entropy.
Man, this journey through the science of extreme longevity completely shatters the paradigm of how we view aging.
It really turns it upside down.
We started with the assumption that aging is just this ticking clock, an inevitable rusting of the machine dictated by a rigid genetic blueprint. But we've seen that the genetic code is highly malleable. It's constantly waiting for instructions from the environment.
Waiting for the right signals.
The molecular machines, the telomeres, the mitochondria, the master regulators like AMPK and FXO3, they aren't looking for a magic pill. They are looking for the precise environmental pressures found in the blue zones.
The constant low-level physical mechanical load.
Right, and the mild caloric scarcity that triggers autophagy. The aggressive downregulation of cortisol through deep social integration, and the protective genomic shielding of eudaimonic purpose.
The biology of longevity is fundamentally a biology of connection and mild, consistent challenge. It is the realization that cellular health is not an isolated individual pursuit, but a reflection of the social and environmental ecosystem the organism inhabits.
And this leaves you listening to this with a truly vital question about the architecture of modern life. We organize our entire adult existence around the financial pursuit of a concept called retirement.
They do.
Our cultural ideal of retirement is a sudden, total cessation of demand. We stop working, we reduce our physical load, we isolate ourselves in comfortable enclaves, and we embrace a life devoid of mandatory challenge. We view doing nothing as the ultimate reward.
It's the goal for so many.
But if the molecular gerontology we've discussed today tells us anything, it's that the absence of demand, the removal of meat, the loss of the moai, the sudden evaporation of ikigai, is interpreted by the genome as a signal to shut down the machinery of repair.
The cells think they aren't needed anymore.
So is our modern, sedentary, isolated concept of retirement actually a biologically lethal construct? What if the key to living to 100 isn't finally earning the right to rest, but rather ensuring that you have built a life where you are perpetually deeply required not to?